Welcome back friends. Welcome to my channel. Here is new video for the development biology, and today I will discuss about the embryonic induction. Embryonic induction is not so simple process. It has many example and complication in later to that topic for CSIR and geology or other life sciences classes. So I will make this video in parts. I will try to explain all the aspects of the embryonic induction in parts. So in one video it is not possible. So in this video I will tell you introductory pattern for this. What is the embryonic induction? First of all, embryogenesis is the formation of individual from zygote. You know it very well. What is the zygote? Diploid structure formed by male and female gamete and fused on the zygote. These are haploid, and diploid. If I talk about mammals or human, the male gamete are the sperm, ovum. Sperm fertilize the ovum and form a diploid structure, that is the zygote. Later on the cleavage starts. After that cleavage occurs and the cleavage form the multicellular. Single cell formed into the multicellular structure. So here the multicellular structure is formed, that is called morula. After that it is called blastula, then gastrula. You know it very well. Later on the organogenesis occur, then the offspring is formed. So this is very integrated and complex structure. So here, what is the induction? During the cleavage, it is a special type of division from the normal mitosis. It is called as mitosis, but is differ in many aspects. I will tell you in lecture of the cleavage. So firstly I want to discuss here the embryonic induction occur from the cleavage up to the embryogenesis, up to the offspring formation. So what is its meaning? I hope it is clear to you. What is the meaning of the embryonic induction? Embryonic induction simply means induction. Induction means instruction, giving instruction to something. So here in the embryonic life, embryogenesis, one cell, that is the cell 1, sends signal to the cell 2. Cell 1 sends signal to the cell 2 and cell 2 form the specific structure, protein or other entities. What is its meaning? Without the effect of cell 1, cell 2 cannot form these molecules, these structures. This is what I call the induction. I hope it is clear to you. The cell which influences the other cell, it is called inductor cell, and which cell received that signal is called responding cell. This is called responding cell. And there are some cues, chemical structures of protein that are secreted in the vicinity by cell 1 and they diffuse inside the cell 2 and influence the environment of the cell 2. These molecules are called as inductor molecules or evocators. I hope it is clear to you. The cell which sends the signal is called inductor cell, and which receive the signal is called responding cell. And the ability to conceive that signal, ability of the responding cell to receive that signal, what I call here is the competence. I hope this fundamental idea is clear to you. This is all I call the embryonic induction. Let's talk more about it. Types. Embryonic induction can be categorized into two types. First, primary embryonic induction. Second is the secondary embryonic induction. Primary embryonic induction is the fundamental processes. First of all, the primary embryonic induction occurs, and it occurs in the blastogenesis, gastrogenesis, and this occurs after organogenesis, during organogenesis, also after the delivery. So, primary embryonic induction means the formation of primary neural axis, such as neural tube, then brain, then spinal cord. This is the formation of the embryonic structure related to the nervous control. First, the nervous control is formed here. And the secondary induction is the formation of organs, metamorphosis of them, proper formation of offspring. So, this is the type and meaning of primary and secondary embryonic induction. I hope it is clear to you. So, this was the fundamental behind the induction. So, there come the experimental evidences, examples related to primary and secondary embryonic induction, and the macromolecular cycles, the regulation of protein that form the full development of organism. What does its meaning? So first, if I talk about the primary induction, what come under it? First of all, regional specificity of primary embryonic induction. This is first clue. Then experiments which prove primary induction. What does they prove? These experiments prove the pattern in which the primary induction occur. Also, here come under the primary induction theories. Theories of primary embryonic induction. So these are very complicated things, and I will discuss later on about them. And in the secondary induction, we will follow same secondary embryonic induction. It is commonly called in many books epithelial mesenchymal induction. Epithelio-mesenchymal induction. Mesenchyme layer is the below the epithelium, and it is a very important layer, send the molecule which help in division and formation of structure from the epithelium. So this is also called secondary induction. Also here regional specificity, types of secondary induction, they will also come in here, and examples are many here: ectodermal, mesodermal induction, ectodermal, endodermal induction. I will tell later on about it. So these examples may include the formation of chick, limb, eye lens induction, and many examples. So we will discuss in detail about them. So firstly I want to talk to you here primary embryonic induction. What we will do here. Here some of the embryologists who gave the experimentation, and we will discuss those experiment and what they drive the conclusion, we will here tell you about them. So in this video I will only tell you name of the experiment and biologist which they have given. So here first very fundamental is the Spearman or Spearman experiment. This is fundamental experiment. Spearman experiment. After that we will tell you another experiment, that is the Saxon and Tobin. Saxon and Toivonen. Saxon and Toivonen experiment give the regional specificity of the primary embryonic induction. After that, another experiment we will discuss here. The name here is the Mangold experiment. This is very important experiment, comes under the primary induction. Then sandwich hold flatter. This is very important. Hold flatter sandwich experiment. We will discuss here, and after that we will discuss another very important experiment, that is the given by Smith experiment. After that we will reach up to the Newcoop center, and then we may capable of defining the actual pattern of the primary induction. So this was all about here, and from my video you could have aware from the meaning of embryonic induction. So if you like this video, share this video among the CSIR net aspirants and biological students. And this is equally important for all from fundamental +2 classes, BSc and MSc in the geology, life sciences, experimentation. So I hope it will helpful for you. In the next video, I will discuss in detail about the primary induction. Thank you very much and good luck.